Okay, today's topic is involving walking. The first question is walking the audience through how GABA works as an inhibitory neurotransmitter. Then the second question, describe how the knockdown of GABA vigabatrin will lead to the prevention of seizures. Then the third question is obviously showing the 3D model of vigabatrin mechanism. This is a specific program. All right, the first question talks about. Okay, I'll start off with saying GABA stands for gamma aminobutyric acid, which some people often call the molecule for relaxation. GABA is a neurotransmitter that is located in the central nervous system. It reduces the activity of the neurons that it binds to. It also inhibits the nerve transmission in the brain and is the calming of the nervous system activity. It calms it down. The nervous system is made up of an individual cells called neurons. These cells are sort of, people call it the body's wiring. The nerve signals are transmitted through the length of neurons as an electrical impulse. When a nerve impulse reaches the end of the neuron, it can jump over to the next cell using chemical messengers, which are called neurotransmitters. In the cell's nervous system, which is mainly made up of the brain and spinal cord, neurotransmitters pass from neuron to neuron. Neurotransmitters are stored at each end of the neuron, and when the neurotransmitters reach the neighboring neuron, the neurotransmitters get attached to the specialized receptor sites as like a lock and key method. When enough neurotransmitters attach to the receptors, the neuron fires, which sends an electric impulse down the neuron's length. Um, GABA is made in the brain cells from glutamate. And functions as an inhibitory neurotransmitter. This means that it blocks nerve impulses. Glutamate acts as an excitatory neurotransmitter, and when bound to adjacent cells, it encourages them to fire and send a nerve impulse. GABA does the opposite and tells the adjoining cells not to fire, and it says not to send an impulse as well. So obviously the calming effect. The The second question addresses. The second question is describe how the knockdown of vigabatrin leads to the prevention of seizures, and that is as first. Let me first define a few things. Epilepsy is caused by the degradation of GABA, which is an aminobutyric acid. Degradation of GABA levels in the brain. GABA ATs, which are aminobutyric and acid aminotransferase. Selective inhibition raises GABA's concentration, releases GABA's concentration in the brain. The anti-epilepsy drug vinyl GABA vigabatrin target is GABA AT and it is a PLP dependent enzyme. This mechanism of inhibition allows the formation of the enamine, which is the dissociate from the PLP, which gives an enamine to yield a stable complex at the active sites, which often leads to an irreversible inhibition of the enzyme. This is why vigabatrin is called also newly called the anticonvulsant drug. Um, and the mechanism, I'm going to show a chart after this, and the mechanism that I'm about to show on the next on the image involves the azellylic isomerization followed by the proton removal, as in the normal catalytic mechanism with substrate. Um, Not like GABA. This isn't like GABA. Vigabatrin is converted into an electrophilic species, and which can undergo the Michael addition at the terminal carbon of the vinyl carbon group, vinyl of the not the, the vinyl carbon of the vinyl group by the active sites LIS 329. And to give two, adduct two can easily be tautomerized to three. And then the actual inactivation is adduct, which is or4. And the structure for that I have right here in this Google Doc. As you can see, there we go. Shows the process I was just talked about. And 1, adduct 2, adduct 3, and adduct 4. And then this is a 3D structure of Vigabitrin, as you can see, the structure. And thank you for letting me present today on this topic.